Æonome, a secret hidden for 3 million years. LIPOTRUE is proudly launching a new bacterial ferment with a novel anti-aging activity working on the skin's microbiome. Æonome, an AGEBIOTIC that offers a new approach to the skin's microbiome. The microbiome world is a complicated and still yet to be fully understood world. LIPOTRUE aims with this product to explore and fulfill the microbiome trend, helping brands achieve claims around this area that are easily understood and in high demand by customers, which is what I will be explaining to you in this presentation. Sorry, I'm struggling with this. My apologies. I was at the end of the presentation. So, carrying on, Æonome is a fermentation of a Bacillus species that is found in the underwater cave discovered in the island of Mallorca, Spain. This cave has been underwater for over 3 million years and is until now has not been accessed by any humans. This cave microclimate harbors unique microbial communities, and these microorganisms that live on this cave contribute to the formation of biominerals, stalactites and the stalagmites that you can see here that shape the inner feature of our cave. The Paz de Valguarnera cave was discovered over 50 years ago in 1968, from ground to underwater levels in labyrinth galleries with a length of over 74,000 meters. This unique cave has exclusive access to research scientists only. The microorganism, or one of the microorganisms found in this cave, was isolated, and the bacterial ferment extract was obtained to give birth to Æonome. Ion, or Ion in Latin, means vital force and eternity, which comes from the ancient Greek of god of the ages and time, Aion, also known as Cronus, which is why we see this gold-colored theme all around the presentation. Ion, also a term used by the philosopher Plato in the myth of the cave to denote the world behind the perceived world. And that's what the analogy that we are doing here. The skin microbiota, with more than 7,000 species, is the unseen world behind the secret for youthful skin appearance. Æonome unlocks the secret of ageless or anti-aging properties of the skin microbiome. The microbiota is the skin's first line of defense. It's made of complex ecosystem of billions of microorganisms such as bacteria, fungi, yet to be fully discovered and understood, just like the labyrinthic chambers in Æonome's cave. One thing we know for sure, an alteration to this symbiosis and equilibrium within the microbiota may lead to dysbiosis. There's some definitions in the world of the skin microbiota that we already know, and they're known. We have prebiotics. They are a substrate or nutrients for the microorganisms. We have probiotics, which are alive, helpful microorganisms. We have postbiotics, which are metabolites from microorganisms, or meaning what they excrete. And a novel concept now being introduced by LIPOTRUE, AGEBIOTICs. An AGEBIOTIC is a probiotic that is metabolized by the commensal cutaneous microbiota, enhancing the release of ageless postbiotics, meaning nutrients for the skin microbiome that induce the release or the synthesis of ageless or anti-aging properties by the microorganisms. Here we can see a drawing of our skin and its main components with the shape of a stalactite, linking the origin of the active and the main applications on the skin. To first assess the efficacy of Æonome, We used an in vitro model of colonized skin using human epidermal keratinocytes in a combination of 5 microorganisms that represent the most abundant and culturable species found on healthy facial skin. The Staphylococcus epidermidis, the Staphylococcus hominis, the Cutibacterium acnes, the Corynebacterium cerosis, and the Malassezia restricta, which is actually a fungi. A summary of the efficacy testing we have performed is seen in here, in vitro and later on in vivo. Æonome stimulates the growth and maintains the bacterial diversity balanced, reinforcing the skin barrier. Reinforces the integrity of the colonized keratinocytes by the synthesis of keratin, filamin, tubulins, wave and ERM complex, leading to skin integrity reinforcing. It has an anti-inflammatory efficacy shown by the increase of annexin 1, decreases epidermal junction flattening by stimulating collagen 7 in vinculin. Overall, it increases the skin age resistance by reducing and protecting cells from oxidative stress, increasing the production of antioxidant metabolites produced by the endogenous skin microbiota, and has this anti-aging efficacy with the synthesis of ageless proteins. We now start with our efficacy testing with our first in vitro test. We can see here the evaluation of the antioxidant effect of Æonome by analyzing the synthesis of reactive oxygen species, ROS, produced by keratinocytes when exposed to hydrogen peroxide and incubated with Æonome for 24 hours. So oxidative stress can be chemically induced in vitro by exposing keratinocytes to hydrogen peroxide, which causes irreversible ROS production. In this study, we have evaluated the potential protective effect of the bacterial ferment against hydrogen peroxide-induced ROS production in an in vitro model using human epidermal keratinocytes. On the right-hand side, we can see how Æonome, tested at 1.5% and 3%, is able to protect the keratinocytes against oxidative stress by decreasing the production of ROS up to 23% less than control. We used ascorbic acid here as a positive control, as you can see on the graph here in brown. On the left, measuring using qPCR, we can see the increase or the upregulation of the expression of the genes catase and ferritin on oxidative stressed cells. These are known for stimulating the natural detoxifying mechanisms of keratinocytes. Catase is a key antioxidant enzyme involved in the body's defense against oxidative stress, and ferritin is a major intercellular ion storage protein. We can say that Æonome protects directly the skin from oxidative stress. Here you can see the results of our in vitro model of colonized skin and how Æonome promotes the contribution of beneficial microorganisms, strengthening the skin's microbiotic barrier. We use the same model with the 5 commensal skin microorganisms representing a healthy skin microbiome. And we incubated epidermal keratinocytes for 8 hours, colonized with this commensal skin microbiota, and measured its values with and without 3% Æonome. The first bar in the graph represents the individual contribution of each strain in the beginning of the treatment. The second bar, after 8 hours of treatment, the strains decreased in numbers, while C. cirrhosis contribution gets more prominent. And the third bar, after 8 hours of treatment with Æonome, we can see a moderate and balanced increase of the mild commensal microorganisms, more prominently the Staphylococcus epidermidis and hominis. We can say Æonome is having a probiotic activity on the skin's microbiome, so it's providing the microbiome the correct nutrients to maintain this symbiotic equilibrium within the medium. In these pictures here, we can see the fluorescence and electron microscopy images of the cell structure of keratinocytes used in the previous test. We look at the morphology of the cell structure or wall, and we are able to see on the bottom image on the right-hand side that colonized keratinocytes incubated with Æonome maintained the integrity of the keratinocyte structure versus the non-incubated with Æonome, as seen in the middle right-hand side picture, when compared with non-treated keratinocytes. what I can say is that Æonome acts as a shield to restore epidermal skin morphology. We now check the biological activity that the treatment exerted on the skin microbial representatives. First, we check the ferric reducing antioxidant power, or FRAP, of the supernatant of the in vitro cultures of colonized keratinocytes treated with 3% Æonome. We can see in this graph how Æonome increased the FRAP by 7.57 fold when compared with untreated colonized keratinocytes, seen in the blue bar. The benchmark used as a positive control here was quercetin, the flavonoid known for its potential antioxidant activity, and Æonome showed a 2.7-fold stronger activity than this benchmark. The antioxidant power of Æonome is seen by the increase of antioxidant activity through its metabolization by the skin microbiota. In here, on the top rectangular, we see a schematic of the interaction of Æonome and the microbial community and how he stimulated these strains to release antioxidant metabolites. So this is a novel discovery. For the first time with Æonome, we prove how a novel bacterial ferment is able to increase the antioxidant power of skin microorganisms and identify specifically the metabolites or postbiotics that are being released by them. The supernatant was analyzed by metabolomics using LC-MS for the specific identification of antioxidant metabolites involved in the FRAP antioxidant activity. We can see here the three specific metabolites identified. We identified coumatic acid, a powerful antioxidant, is described to be even more potent than vitamin C. We got tetrahydroisoquinoline, which is an antioxidant alkaloid. It's also described to be very potent and mostly produced by plants and other microorganisms. And aspartyl isoleucine is a dipeptide, is also known for its antioxidant properties. This is a big highlight of this project, since we prove the relevance of the skin microorganisms on the protection of our skin's health through antioxidant mechanisms. And we complete here the final picture of the mechanism of action of Æonome by studying its effects through the microbiome on epidermal keratinocytes, in this time using proteomics. Using the same in vitro model of colonized skin, the treatment with Æonome for 20 hours showed the upregulation of key ageless proteins by the primary keratinocytes. On the graph, we are showing the fold increase of the different protein founds, Related to cellular cytoskeleton reinforcement, we found rhodopsin and myosin as part of the ERM protein complex with filamin and vinculin. All of them interact with the actin filaments of the cells. We also found the upregulation of tubulins, also related to cell integrity. We observe an increase in the proteins involved in the dermal epidermal junction improvement, such as collagen 7 and keratin 79. And the two novel addition proteins with antioxidant properties. We have the SOD1 and one with anti-inflammation properties we found was the annexin 1. And this is a drawing of a cytoplasm of a cell showing the location of the proteins, the newest and the already known proteins identified in the previous slide, and the relevance on the key processes of structural integrity, as we said before. So we move on to our clinical tests. We have done these in a slightly different way than standard. We used a panel of 40 volunteers, all of them with healthy skin, aged 40 to 55 years old. And in this case, half of the panel used the cream in the whole face containing 3% Æonome, and the other 20 volunteers used the placebo cream. At the beginning and after 28 days of treatment, a cotton swab was taken off the volunteer's forehead to perform next generation sequencing evaluation. Here we show the results for wrinkle reduction by fringe projection at 7 days in the crow's feet area. The wrinkle area decreased in 8% in 7 days, and in volume we see a decrease at 17.4%. Looking at other 3D parameters of wrinkle measurements at 7 days of treatment, we look at the maximum wrinkles amplitude, or ST, and saw a decrease in almost 4%. We measured the mean difference between the peaks and the values of the wrinkles, or STM, with an observed reduction of 2.5%. And finally, the rugosity average, or SQ, we observed a decrease in 4.3%. And as you can see here, all measurements are increased with the placebo. So we're definitely seeing an effect of Æonome here. And some of the photos here, some of the visuals by fringe projection of the crow's feet area of a 50-year-old volunteer after the 7 days of treatment. We observe a reduction of 22% in volume of the main wrinkles. Some more visuals here, and we can see here pictures of the forehead at 28 days, up to minus 27% rugosity decrease over time at 28 days in the forehead. Here we can see the forehead area wrinkle results, a decrease in up to 85% of wrinkle area and an up to 28% reduction in overall rugosity of the skins at 28 days. And always best to see photos, and very visual here, the photos we have of the different volunteers, one of 41 years old, another one of 43 years old. We also measured the moisturization of the volunteers' skin and found that Æonome helped improve the hydration of our volunteers with a short and a long-lasting effect. And this slide highlights the effect of Æonome on the skin microbiome and links really nicely the clinical studies with our in vitro results. As I said before, for each of the volunteers, a swab sample of the skin's microbial surface of the forehead was collected before and after 28 days of treatment. No volunteers were showing any type of dysbiosis, as this new active is meant to be used on broad spectrum skin types. We used the next generation sequencing analysis to identify the operational phylogenetic units of more than 7,000 microbial species that were found present on the skin of the volunteers. The main results obtained showed that the relative abundance of these species were not altered by Æonome in consonance with healthy skin microbiome, which is able to balance naturally. And as a plus, Æonome showed a modulation of the relative abundance of key relevant microorganisms as observed in the in vitro model we used before. There was an upregulation of the S. hominis and Corynebacterium species, important on moisture areas and described to release antioxidants, which is what we saw. There was finally a trend by Æonome to maintain the contribution of the Staphylococcus epidermidis and decrease the C. acnes, slightly increased here in the placebo. As a result of this balance and modulation of key species, Æonome links nicely its in vitro activities with the clinical results observed here. And on this slide here, we can see in a very visual way how Æonome, produced by Bacillus Ferment, collected on a protected subterranean ancient cave, is capable of unlocking, as you can see here from left to right, the natural process in which your microbiome, through the release of antioxidant metabolites, balances, nourishes, protects, and takes care of our skin to rejuvenate and improve its quality. And as some of our technical data here, so it's Æonome, as we said, it's a marine fermentation. Our recommended dose is 1.523%, and is 100% natural according to the ISO standard 16126, is Cosmos approved, and is also 100% readily biodegradable. It comes in a solution, it's water soluble, and is completely preservative free. And some of the possible applications that we could suggest for Æonome products supports the unseen world of microbiota, broad spectrum microbiome ageless product, AGEBIOTIC for an anti-aging efficacy, or soothes the skin by reducing oxidative stress, slow beauty, and short efficacy. So are you now ready to enter the microbiome world with Æonome? I hope so. Thank you very much for all of you to attending the seminar today. We understand there are very challenging situation right now, but we hope soon this will be a memory. And all our teams are available to support you in any way you need. And if you would like to ask me some questions, please go ahead.
Okay, thank you very much, Raquel, for the clear and interesting presentation.
Thank you.
Grazie anche a tutti i partecipanti al seminario. Se qualcuno avesse domande o curiosità, non esitate a riattivare il microfono e a esporre le vostre domande.